Hi everybody, my name is Anastasia, and today we're going to be talking about cells. So we're going to be looking at a PowerPoint with some pictures, some information, just again to give us an idea of what it's like to be living. We're going to look at the differences between prokaryotic and eukaryotic, and just going to get some other information. So let's see if I can get back. Here we go. So for my cell cumulative video. So the first thing that we're going to touch base on is how to know if something is living. So there's lots of different ways that we can tell if something's living, but sometimes it's best to just narrow it down to just a few key ideas. So the first thing is that it's made of cells and has DNA, so it has that genetic information to pass on to offspring. It is able to maintain order inside their cells and their bodies. They can regulate their systems. They respond to signals in their environment. They transfer energy among themselves and between themselves and their environment. They grow and develop. They reproduce in order to pass on copies of their DNA. And they have traits that evolve over time. So cell theory. So there's three main components of cell theory. So when we're thinking of cell theory, we have to keep in mind that all living things are made up of cells. The cell is the smallest unit of living things, and all cells come from pre-existing cells. So all those are, so when we are looking at cells, those are the three things that we're going to be focusing on. So cell theory was actually proposed by Schleiden and Schwann in 1838, but can be traced back to the 17th century. Over the years, advances to the microscope allowed for better observations of cells. Cell theory built the foundation for understanding the structure and function of cells, organisms, and diseases. So although this has been around since the 17th century, it's had a lot of modifications, a lot of things that have changed with technology and advances in science. So it's important to keep in mind that there wasn't always just straight out of the box, that this was something that did evolve and change and get modified. So looking at the types of cells, so one of the main types of cells that we would focus on is called a eukaryotic cell. So eukaryotic cells have characteristics such as membrane-bound organelles. They're structurally very complex. So the eukaryotic cells have a lot more to them than a prokaryotic. They can be divided into 4 different categories. So there's Animalia. They begin life as a zygote, includes slugs, worms, cats, dogs, lizards, fish, etc. So it really kind of makes us go back and think that even though we're, of course, physically quite different than a slug or a worm, we actually have a lot of common things. There's plantae, so their plants have cell walls made of cellulose. They reproduce asexually by mitosis and start life as embryos. So the third one is fungi. They are not photosynthetic like plants. They get nutrition by breaking down and digesting dead matter. And then the final category is a protista. Everything else that is eukaryotic is placed into this category. It is so diverse that the organisms in this group could be separated into 11 different kingdoms. It's interesting to think that there's only 4 categories when there can be so many different cells placed in protista, but there really isn't a category separated for them. They kind of end up being the dumping ground for cells that don't fit in the other categories. So it makes one think if how many categories we really would have if they actually did separate them and decide to give each cell in the protista their own category. So I'm looking at organelles found in eukaryotic cells. So this isn't all of the organelles, but this is just kind of a sum up of some of the major organelles that we would be looking at if we were focusing on a eukaryotic cell. So the first one being mitochondria. So it gives the cells the energy needed to move and grow by breaking down food molecules. The second one is the Golgi apparatus. It's The products produced by the cell are chemically tagged and packaged and sent out to the other organelles. The biggest way to, the most common way I think that we think of the Golgi apparatus is kind of like the post office. So they get all the information coming in, it gets organized and shipped out where it needs to go. The third one is endoplasmic reticulum, or ER, series of roadways that connect the nucleus to the cytoplasm of the cell. So there's rough ER, which has ribosomes on it, and smooth ER that does not. So then moving on to ribosomes, they're found in the cytoplasm of the cell. So eukaryotic cells have kind of like a gel inside it, and so the ribosomes are just kind of sit in the cytoplasm or the gel. Instructions for protein are copied from the DNA and made into messenger RNA. The fifth one, lysosomes, they're formed by the Golgi apparatus to clean up the cell, contain digestive enzymes. So lysosomes are really important so the cell doesn't get clogged with stuff that it doesn't need. Number 6, the nucleus. Of course, one of the most vital parts of the cell, contains the DNA separated from the cytoplasm by the nuclear envelope. So, like I said earlier, you have the cytoplasm that's kind of like the gel that everything sits in, but the nucleus, to keep the DNA safe and in form, is protected by the nuclear envelope, so it keeps it separated. So I found this picture from galleryhip.com. So it's a structure of a eukaryotic. So as you can see, it has some of the major organelles that we discussed in the last slide. So you have the ER, the endoplasmic reticulum. You have the nucleus, of course, in the middle. The cytoplasm, which is all the white that it sits in, all the organelles sit in. You have the cytoskeleton, all the little dots, I think, that are kind of hard to see, but those are the ribosomes. You have the mitochondrion. So you have the mitochondrion right here, kind of the kidney shape. The lysosomes are like the little, they kind of look like oranges. And the Golgi body is right here. So the Golgi apparatus. So the Golgi apparatus right here, so you can kind of see it has all the canals. And then the nuclear envelope, and then the nucleus. So looking at prokaryotic cells, so organelles that are found in prokaryotic cells, you have the plasma membrane, which kind of forms a barrier around the cell and a rigid cell wall to provide support. The DNA is genetic material found in the cytoplasm and the nucleoid. Ribosomes make proteins in the cytoplasm, so again, with the cytoplasm, kind of like the gel that everything sits in. The flagella, thread-like appendage that allows these cells to move. So it's located on the outside of the cell. It's I kind of think of a caterpillar that has legs, so it allows for movement. The pili allows the cells to attach to other cells. And then the capsule offers another layer of protection, holds moisture, and helps the cell connect to other cells. So the capsule is like the very outside layer of the prokaryotic cells. So prokaryotes break down food using cellular respiration and another type of metabolism called fermentation. They have a simple structure because they do not have internal organelles like eukaryotic cells, so they're much more simplistic in structure than a eukaryotic cell. So looking at the picture from experiment.com, so it has a lot of the organelles that were listed in the last slide. So you have the capsule, which is like the very outside, then the cell membrane, then the cell wall. So they have 3 layers of protection that keeps the cell structured. So you have the capsule, the cell membrane, or the cell membrane, yes, cell membrane, the cell capsule, the cell wall, the chromosomes, the plasmid, the cytoplasm, of course, that fills it, and then on the outside you have the flagellas and the pili. So when we're looking at the difference between prokaryotic and eukaryotic, they have some common factors, but then they also have quite a few things that make them unique. So one of the first things is eukaryotic cells are much more complex than prokaryotic. So they have much more, they have a lot more organelles than what the prokaryotic cell has. The second one, prokaryotic cells have a pili and flagella on the outside of the cells to assist in movement. Number 3, prokaryotic cells do not contain a nucleus or membrane-bound organelles. Number 4, eukaryotes divide by mitosis and prokaryotes divide by binary fission. So that's quite a big difference. Prokaryotes have no cytoskeleton. Prokaryotic cells contain bacteria and algae, where eukaryotic cells are found in animals and plants except algae. So it's interesting to see that it's really based on the type of cell it is. So if it's going to be bacteria and algae, it's going to be more of a simplistic cell compared to what we as mammals and plants have. In eukaryotes, the nucleus is surrounded by nuclear envelope. Prokaryotes have cellular DNA, where eukaryotes have linear DNA. And eukaryotic cells are bigger in size compared to prokaryotic cells. So because eukaryotic cells have so many more organelles and so much more going into the cell, it's going to be quite a bit larger than the prokaryotic cell, which is more simplistic. So moving on to look at viruses. So a virus is a non-living structure that is classified as an infective agent. They are made up of nucleic acids, genetic materials, a capsid, protein coat, and some have a lipid bilayer known as an envelope. They are not considered cells because they do not have a metabolism and cannot reproduce on their own without another cell to latch onto. Viruses infect hosts in order to reproduce. So that's the only way that they're able to reproduce and to divide is if they have a host that they can latch onto. Viruses infect cells in 2 ways. So the first way is through the lytic cycle. They inject DNA in the bacteria and make copies. And the other way is through the lysogenic cycle. They inject into the cell, which becomes part of it, and the cell copies the virus. So it actually is injected into its DNA, and it becomes part of the DNA and is able to divide without the cell knowing that it's there because it's just dividing. It's doing its usual function. So this is just a picture of what a virus would look like. So moving on, what is a prion? So prions are proteins that exist throughout the body but mostly found in the brain. Prions are really quite an interesting structure because scientists aren't exactly sure what they do. They believe to play a role in protecting brain cells from injury. Prion cause disease because they can change shape spontaneously, which is really quite interesting that they have that capability. Normal protein changes shape and meet an abnormal prion. The normal protein will actually change shape and the result will be 2 abnormal prions. So in the video that we watched earlier in the semester, it was really interesting to hear the doctor talking about how the spoon, so you have a spoon and you have the spoon and the fork, and when they meet, they actually both become spoons, and then they move on and copy and infect, which is really quite interesting. So prions cause diseases such as mad cow disease, Jacob disease, fatal familial insomnia. So the prions can lay dormant in the body for 15 to 25 years before any symptoms are experienced. They are not alive, they cannot be killed, and there is a way, but you can actually inactivate them, but there's no way to actually get rid of them. So over the course of this module, we've had quite a few discussions about different scientists and their discoveries. So who to thank? Although there have been an array of scientists discussed in this unit, the one I would like to thank is Carolus Linnaeus. He lived between 1707 and 1778, and we owe our thanks to him for introducing the hierarchy of how organisms are classified. He introduced to scientists the kingdom, phylum, class, order, family, genus, and species. Although some of his ideas have been adapted to fit the information we currently have, he was the founder of this organizational system, which is really quite interesting to think that someone was able to come up with this, because it just makes it so much easier for us to understand how animals and plants are related and trying to understand their origins. And that is it. And I hope that was helpful, and I hope that provided a lot of information. Thank you so much.